So today we have Mark Daly, who is the chief of the Analytic and Translational Genetics Unit at Mass General, an institute member at the Broad Institutes, and co-director of MPG. And he'll be talking about Introduction to Complex Trait Genetics. Thanks, Mark.
Thanks, Chris. Let's jump right into it. As Chris said, I'm Mark Daly. Nice to see all of you here. I'm going to give a very brief and historical overview of human genetics, particularly with respect to how we study and find the genes for complex human diseases. Now, I think we all have known, for all of our lives and have all grown up in being trained in genetics, thinking about the heritable diseases, so heritable rare diseases, cystic fibrosis, Huntington's, muscular dystrophy, and so forth, where it has been, for more than a century, obvious that many of these types of conditions are strongly heritable in as much as they run very, very much, in families. And if you have a first degree or even in some cases second degree relative with these diseases, the risk that you have to these diseases is very, very much higher than the general population. Now, as it turns out, common diseases also, run in families, and you can make heritability estimates. Here's some, numbers for lifetime risk to type 2 diabetes, perhaps, given in a certain population. The numbers for many other common diseases, neuropsychiatric diseases, immune-mediated diseases, are even more profoundly different, from identical twins back to siblings, back to neighbors. And what this tells us is that inherited variation in your DNA, the DNA that you're born with in your germline, influences disease risk. And the whole premise of what we've been doing in medical and population genetics here for the past, more than 20 years, is trying to use the genome to provide fundamental clues and insights into the underlying biology of human disease. The eventual goal is, of course, that those fundamental biological insights, which are causal links to human disease, ultimately, when we understand them, would provide us novel avenues for therapeutic hypotheses that might be effective and would really be geared at the root causes of disease rather than the symptoms. So, putting that another way, we're not very good at developing drugs as a biomedical community unless we can start with credible hypotheses that targeting a certain gene or a certain pathway is very likely to be efficacious in this disease. And we've invested tremendous amounts over the years in many, many therapeutic programs that, based on a flawed initial hypothesis, no matter how good you do your chemistry, no matter how accurate and true your animal model or your cellular model of disease is, will not result or cannot result in an effective therapeutics. And so we lose the vast majority of late-stage therapeutics in large part due to a lack of efficacy because the initial target hypothesis was incorrect. We also lose quite a number for adverse responses as well. So finding and understanding the genes and mutations that underlie disease, in many cases, might be one of the most reliable opportunities we have to gain that fundamental biological insight. In some cases, for example, in neuropsychiatric disease, it may be one of the only ways in which we have an opportunity to gain that biological insight, because unlike cancer, there's no tumor to cut out and study. Unlike the inflammatory bowel diseases, we don't have access in live patients to the fundamental cells of the immune system and the fundamental cells of the gut that we could then study and understand the disease process. We have to start in certain diseases, for example, the psychiatric ones, with almost no biological knowledge or ability to access the relevant cells and tissues, even if we knew what they were, in live patients. So genetics offers us a good deal of hope. Now, we were all, of course, trained in what we might describe as first century genetics, and the groundwork of how we discover genes in the late part of the last century was really all constructed in far, far in advance of even understanding the structure and chemical makeup of DNA, and even understanding what a gene was and what a chromosome was. Mendel's laws, the first genetic maps that were constructed based on the relationships of phenotypes and how they are transmitted from generation to generation in flies originally, and really the principles by which inheritance and linkage analysis work were all developed in a 100-year period that was far in advance of our ability to molecularly study and understand DNA. And why this became such an active field was, as I mentioned earlier, it was clear that many diseases actually traveled predictably and consistently through families. And these are diseases that we think of, or that we grew up thinking of, as genetic diseases. And the tools that we developed in the 70s and 80s were very effective, and then molecular techniques, as they caught up, particularly in the late 1980s, began to be very effective at being able to identify the specific region of the genome, and then through a lot of hard labor, wrestling gene by gene, ultimately the discovery of mutations and genes underlying a number of severe, inheritable, and highly penetrant diseases. And this strategy of tracking disease inheritance through larger and larger families and tracking down ultimately the specific responsible gene was effective because in many diseases there is a deterministic relationship between genotype and disease state. So you will get CF, and you will only get CF if you carry 2 mutations in the CFTR. And so that relationship is a very simple one mathematically, and it inherently breeds true in larger and larger pedigrees. So you can track these things over many generations, and it actually became quite, straightforward and routine to do so throughout the 1990s, as I'll show in a minute. Now, as it turned out, common diseases are also highly heritable, and things that even have a lot of diagnostic complexity to them, like autism and schizophrenia, are among the most heritable entities that we study in this building, no matter what floor you're on. But despite developing those strategies in the 80s and 90s to find these single deterministic genic causes of disease, we applied those in a much larger scale to autism, to schizophrenia, to diabetes, to type 1 diabetes, type 2 diabetes, a whole host of cardiovascular, cardiometabolic, autoimmune, and inflammatory diseases, and had next to no success. So the sad little blue triangles down at the bottom, essentially representing a handful of MHC associations to immune-mediated phenotypes, and the ApoE association to Alzheimer's disease. And unfortunately, in reality, this was something that we should have understood, or we should have predicted well in advance of starting our genetic studies in the 90s for more complex diseases, but it had been sort of an overlooked part of the history of genetics. So not that first century nice linear Mendelian thread, but a thread that started more with biometricians around the turn of the previous century, in which it began to be realized that there were many traits in humans that were quite continuous and made beautiful bell curves, but they were also extremely heritable. And this then caused a great deal of consternation for a good 15 to 20 years amongst dour-looking white men who couldn't quite understand how you could have some traits that were continuous bell curves without violating Mendel's laws. And so they went back and forth with this, for a while. And it turned out to be actually plant experiments that first lent empirical insight into what was going on. And these plant experiments, and ones that soon followed in flies as well, were of the flavor that if you take 2 strains of a plant or a fly or what have you, that have a very strong phenotypic difference in one particular area, in this case corolla length, and you breed them, sometimes you discover that very large phenotypic difference is not caused, as it was in Mendel's garden, by a single mutation, but it was actually caused, and you empirically demonstrate this by selective breeding and then picking members from the population, selectively breeding them, and so forth. It was actually caused by the mass action of a large number of independent variants, each of which contributes a small amount to that difference. And how they got so different in the first place, who knows? Probably it was human breeding in the case of the plants. But what this demonstrated was that not all large phenotypic differences in individuals need be caused by a single large acting variant. Fisher then sort of resolved the sort of mathematical debate around this question by simply pointing out that you don't need to violate Mendel's laws if you've got hundreds and hundreds of genetic variants, each of which behave in a perfectly Mendelian fashion, but their Mendelian effect was to increase or decrease height by a couple of millimeters, then you would get exactly what we see in the population, which is a perfect bell curve, which was 80–90% heritable. So in light of that model, the failure that we experienced in the 1990s with respect to trying to track single gene causes for complex and common diseases was actually reasonably easy to understand. First of all, there are many genes that are going to be involved in very complex biological processes. And second of all, we can't expect the mutations necessarily to have sort of an on-off effect with respect to whether you have an MI on your 60th birthday or not. They are impacting in subtle ways the biology of the human body, and along with exposure and environment, place you at higher or lower risk. And so those things can be very, very heritable, but they don't necessarily track in a clean and deterministic way with genetics. So this shouldn't have bothered us, and in retrospect it all makes sense, but we were sort of optimistic in the 90s that, all right, maybe linkage analysis doesn't make sense, but maybe we'll still try it anyway because that's what we had the technology to try. So basically the hypothesis that emerged largely from sort of the empirical experience in the 90s that told us that, okay, single gene mutations are not, by and large, the easy explanation of complex traits, was that we might have to sort of go back 100 years and listen to Fisher and many others who sort of conceived of this, what's come to be known as an infinitesimal model, in his words, that there might be many, many small effects that contribute to phenotypic differences that are quite heritable in the human population. And so one of the ideas that is useful mathematically from this is something known as a liability threshold. Again, most of what we do is at least 100 years old, apparently. What this actually represents is that you can say, all right, in this model of Fisher's, if there are lots and lots of little quantitative effects, oftentimes what we end up with is defining a diagnostic threshold that's binary, like you have diabetes or you have hypertension, based on some underlying quantitative trait. And in the case of diabetes and hypertension, we actually pretty much know how to measure those underlying quantitative traits, and we're better served to use quantitative approaches to study the genetics of those. Now, the interesting thing is that, our ability to measure the intermediate, your blood pressure, your hemoglobin A1C, what have you, and define our disease categories, doesn't change the genetic reality of how complex traits are working. We don't know how to measure an intermediate for schizophrenia or an intermediate for Crohn's disease or all of the different processes and pathways that go into diagnosing someone with schizophrenia. We only see the end state there. The genetic architecture of those traits is not any different than diabetes or hypertension or simply tallness versus height. What's different is our ability to sort of access, and this is an important difference, access those intermediate phenotypes or measurements that would help us better understand what the genes are doing. So, pretty self-explanatory statement. If we knew what the genes were in advance, they'd be easy to find. We spent a period of time as a field recognizing that linkage analysis wasn't working, but in advance of having genomic tools to sort of systematically study the genome, everyone said, well, maybe we can be smart and guess what the genes are. This did not work very well because the whole reason we were doing genetics in the first place was because we didn't have really that much of an idea of what the genes were. And as it turned out, we weren't very good at guessing them. We also didn't really employ very good statistical practice in this era. And so not much came out of it. A few findings turn out to be true, but the vast majority of this effort was not particularly helpful in retrospect. But again, moving, we only move in gene discovery and complex disease in step with what is technically possible in the day. And I think many of you who are younger would be very surprised to understand that in the year 2000, just how little access we had to genomic technologies that could study genomes of patients. We had our genetic studies at this time were, on the order of tens of genetic variants could be studied in a few hundred individuals. And those studies were actually quite expensive. And so in those days, the genetic part was far and away the greatest expense and labor of the study. Phenotypic assessment of patients was, by comparison, not the most expensive part of those studies. The tables have been dramatically reversed in the last 15 to 20 years. So what we had a view of, though, was starting to get a glimpse of what the underlying model should be, would suggest that, unless we were particularly prescient at guessing what the candidate genes and variants were, what we needed to actually do was take a more systematic genome-wide approach. And so we began around when the human genome was being sequenced to develop systematic maps and references of genetic variation that serially came out over the next decade as the first human genome was completing its sequencing. Because obviously what we are looking for in human genetics and medical genetics is what are the genetic differences in individuals that contribute to rare and common disease and human phenotypes. So the first human genome reference is an integral piece of the puzzle, but now, obviously, when we're studying human variation and phenotype, we need to begin to build references not of one human genome, but of ultimately thousands and thousands. And so that's part of what we here at the Broad were very integrally involved in from the earliest days, in the late 90s of the Human Genome Project. It's an effort that continues today with resources that, like EXAC and NOMAD that Dan MacArthur's group is putting together. We continue to, recognize the importance of having, for medical genetics, as comprehensive a set of reference genomes as possible for comparison with our individual patients, for comparison with our case series in different clinical areas. Now, that was all great, but much more important to the progress that I'll highlight during this talk was the technological advances that had to happen. As I said, our studies in the year 2000 were very small, very time-consuming, and just not a lot of fun. And so watching the technology evolve, first array-based technology that allowed us to simultaneously look at hundreds of thousands of DNA variants rather than one at a time in a labor-intensive way. And then following that, the next generation sequencing technologies, which essentially allow us to recapitulate the entire Human Genome Project, which, if anybody was unaware, took more than a decade, cost literally billions of dollars, can now replicate that entire study and many, many more in a matter of weeks for a few $1,000s. So the combination of these resources, and in particular these technologies, enabled us to begin what we describe as sort of the era in human genetics of the genome-wide association study. So with respect to complex traits, we had not benefited really much at all from the first golden age of medical genetics in the 90s, where we were all so tremendously enthused that we could now finally find the genes and mutations underlying severe monogenic diseases. So this last decade has been the period of time where the analogous progress has finally been made in complex traits. So I'm going to highlight along the way type 2 diabetes. Here were examples of what was known in the year 2005. Basically, there were some very early onset monogenic forms of diabetes, similar to where our fields were with respect to breast cancer findings, with respect to Alzheimer's findings. For late onset diseases, we were able to apply the Mendelian strategies that you'll hear more about during the series to very rare and very early onset forms of disease. And it was the early age of onset that allowed the recognition of particular families as having a unique form that underneath the hood would have a single gene cause to it. So this strategy, very effective in gaining us our first genes in Alzheimer's, in various forms of inherited cancers, and in type 2 diabetes, also severe dyslipidemias, but did not sort of carry over into the generic forms of disease and did not carry over into earlier onset diseases. So in immune-mediated disease and neuropsychiatric disease, we don't have any examples of this strategy because it was the very early age of onset that flags families as truly unique in this space. And in type 2 diabetes, there were a few other findings that did come from the candidate gene era that seemed to robustly stand up to the test of time. There were obviously a much longer list of such findings that did not hold up in the fullness of time. And I'll also give highlights from inflammatory bowel diseases, Crohn's disease, and ulcerative colitis, where the combination of linkage and some very, very hard positional cloning work underneath that had arrived at two associations that were, in fact, reliable, but in each case, only a tiny fraction of the heritability, really less than 1% in diabetes and maybe 1 to 2% in Crohn's disease, were explained by those existing associations. So genome-wide association comes along. What is a genome-wide association study? Well, it's basically just a very simple case control association study. So we actually went backwards in time with respect to the complexity of our analyses, just taking a genetic variant and asking whether its incidence is different in cases and controls, something we'd done for a very long time, but now we were doing this on a massively parallel scale, if you were, hundreds of thousands to millions of DNA variants covering the entire genome. So now we're not asking a candidate question, we're just saying, hey, let statistics and genetics tell us where we should be looking, and let's take our hands off the wheel. Now, unfortunately, taking your hands off of the wheel with respect to data generation in biology is always rife with initial challenges. And so the vast majority of the work in conducting a genome-wide association study is actually in the quality control of the data. There's all sorts of different ways in which you can introduce technical artifacts from mixing different DNA sources. You can introduce genetic artifacts by mixing samples from different populations and not controlling for that. And obviously simply needing to automatically track all of the samples very, very precisely and have built-in fail-safe mechanisms for recognizing when things have gone wrong. Because we abruptly moved from a time in which genetic data was collected in a very manual way, running gels, and people intimately cared about the DNA plates and took very good care of what was going on, to a time in which, most importantly, data collected at this massively parallel level you can't review every genotype by genotype. And so in the old days, ultimately we'd all sit around the table and look at the gel and say, oh, that's a heterozygote, that's a homozygote. And this was kind of fun, actually, but you couldn't any longer do this when you were collecting 100,000 or 500,000 genetic variants across the genome. At the end of all of this quality control activity, which is about 95% of the work, testing for association, as I said, is actually reasonably straightforward. And despite a whole slew of efforts to make this process somehow more powerful and exciting and interesting, it still remains pretty much the case that the vast majority of insights that we get from genetic studies come from single variant case control comparisons such as this. We use slightly better regression techniques to control for all sorts of confounders in the phenotype and genotype space, but it's about as simple as observing that, gee, there's a lot more allele A in controls than in cases proportionally, and attaching a statistical value to that. And we were very happy when our first IBD study resulted in some result that looked like it was, hey, that's less than 1 in a million p-value. Isn't that plenty? It turned out that as it turned out, not necessarily was the answer, because in sort of linkage analysis, we had sort of gotten accustomed to log scores of 3 or 3.5 as being significant, because that kind of corresponds to a traditional statistical significance threshold of 0.05 when you consider how many different independent chromosomal arms are being tested in a linkage study. In a genome-wide association study, we're now performing, sort of a linkage study, but using the entire human family and the entire human history of recombination. And what that means is that, if you move from this SNP here 100 kilobases over, those are in fact completely independent of each other, evolutionarily speaking, by and large. And so we're more on the order of performing about 1 million tests. And so you might actually get a statistical result of roughly 1 in a million purely by chance. And so this, led to some, back of the envelope calculations in 1996 that turned out to be very, very prescient and accurate. And when we actually had collected enough empirical data about, variation and recombination and linkage disequilibrium in the human genome, we could then make much more accurate estimates of this, and a number of groups did that. And we came up with an answer that was really, in quite a similar range to the back of the envelope calculation. So we end up sticking with this 5 times 10 to the minus 8 threshold. So that's where it comes from. It's obviously an approximation, but a very effective one, because as complicated as you want to make that calculation, it's not going to be too far off. So first genome-wide associations results started to roll in 2005, 2006. And, we were excited that there were actually some genuinely, seemingly new findings that came from a number of these. We quickly recognized that we had to see replication as a reliable marker of a result being genuine and true, and something we could then now begin to invest seriously in biology. And we came to recognize, and have seen this over and over again, that, the genetic study from, the UK and the genetic study from Italy can't give you a fundamentally different story about the involvement of a gene. If they are inconsistent with each other, then you need to look very hard technically as to whether this is a robust result or not. And now we've begun making these studies more globally. It's almost certainly and much more generally the case that the genetic result that you see as an association in Europe is going to be repeated very, very accurately if you perform that study in East Asia or in Africa, if the genetic variant has a comparable frequency that can be tested. So those individual first genome-wide association studies in Crohn's disease, in type 2 diabetes, and in a number of other traits came up with, a couple of new findings. So anywhere between 1 and 5 or 1 and 10, these are the findings from a series of 4 genome-wide association studies done in Crohn's disease individually in the time period 2005 through 2007. And so this was, on the one hand, all right, this is many more genes and variants than we had known about before. So you say, that's great. On the other hand, when we sort of assessed the sum total of what we'd learned, we still concluded that these were all collectively explaining less than 5% of the heritability, or the less than 5% of why some people get Crohn's disease and other people's don't. So what then came to pass was the sort of third fundamental change, over and above building reference maps, over and above the technological advances that was required for really profound progress. And that was that we sort of set aside our historical, sense that, we're all competing against each other and racing to find the gene for disease X, and rather recognizing that, boy, there's a lot of genes, it's very hard to find them, and our results are very consistent around the world. So what we actually need to do is work together in a way that, physicists and others had, been doing for many, many years. And this has made a tremendous difference in our ability to discover genes and variants simply by applying the same genetic strategy, but doing it and then putting all the data together, bringing everyone around the table to put their brain power into the analysis of it, and then sharing in the credit. Or the way I sometimes, would describe to people who are resistant to this that you can do your own small study and get full credit for discovering 0 genes, or you can participate in this larger effort, and whatever number of genes we find, number of genes divided by number of investigators is greater than 0. So, this makes sense. And so as we moved from those first studies in 2006, 2007 to larger and larger collaborative studies in the field of Crohn's disease, the returns were much greater than just dividing by the number of investigators or the number of studies or even the number of samples. And that's because as we begin to discover, this infinitesimal model of fissures actually is exactly the way that the genetics of complex disease are working. So we might find, with a study of 1,000 samples, we might find 1 or 2 genes. If we move that to 20,000 samples, we find more than 100. And so that the proportional increase is actually even better than linear for a good part of this curve. It continues to be, although it does have to flatten off at some point. And the best part of this is that these results, unlike what we were finding from the candidate gene era, were durable and reliable and consistent. And so everything that we described when I gave a plenary talk at ASHG exactly 10 years ago this month, from this first meta-analysis study that Jeff Barrett was the first author on, are not only genuine results, they're all confirmed, they're all much more significant in today's data than they were back then. So we actually had, somehow, luckily enough, figured out how to get this right. We also began to understand that looking just at one disease by itself doesn't give you a full picture of what's going on. And so we made much more progress when our historical studies of Crohn's disease and ulcerative colitis, which different investigators were driving, came together in a more holistic fashion. And we found some genes for both forms that were shared overlap. And so the numbers, again, increase to now today close to 250, as I understand it from a recent update, loci involved in one or the other form of inflammatory bowel disease. And a similar thing has happened in type 2 diabetes genetics. Now, because type 2 diabetes is a little bit less heritable, a little more common, the locus discovery was a little bit harder, and but it is sort of reaching a similar point and went through exactly the same explosion once the groups that had originally done, there were 3 groups that had done large studies, and together they had evidence of 1 new gene from those studies. When they put those studies together, the numbers began to really explode off the charts. And so this story is repeated in many, many complex diseases. So the answer is then, okay, so what do we do with all this information? There's a whole bunch of statistical information. It's usually not very easy to interpret because it's a bunch of RS numbers scattered around the genome with large and exciting p-values. But most of all, we've validated this idea of fissures, which was a little bit depressing, and to some extent, that the genetics of complex disease is made up of hundreds and hundreds of tiny contributors, each of which contribute only a little bit of the story. So the question is, if is it possible then that this is useful therapeutically because each piece of the puzzle is a very small piece, or look at it another way, this is like at least a 1,000-piece puzzle instead of the kindergarten puzzle with 5 or 10 pieces. And we think that the point of the studies really was to gather biological insight into the phenotype, and we're beginning to do that, though that's that it's in and of itself, as I'll note, is not so tremendously easy. But we're not really overly concerned with the fact that most of the results that we get from the genetic studies are small, in the same way that we weren't sort of overly put off by the fact that the original discovery of statins, which are effective in just about everybody in the population, came as a result of extremely rare mutations in familial hypercholesterolemia cases, or individuals that carry LDLR mutations. So similarly, if we hadn't done that, hadn't discovered statins in advance, we would have, a good 10 years ago with the first GWAS studies, had discovered that there's a small regulatory variant that is immediately acting right on the HMG-CoA reductase gene, which is the target of statins. And so we would have recognized that this might be a target, but we might have been put off by the fact that the genetic variant in HMG-CoA reductase actually is very common. It has only a tiny effect on serum LDLs. But that's not therapeutically exciting, because I guess what's overlooked in that original sort of simplistic argument is that the natural genetic variant that happens to have arisen in evolution and happens to have been benign enough to survive, tens of thousands of years of evolution, happens to be benign enough that it doesn't do anything bad during embryonic and fetal development and childhood brain development or what have you. None of that has anything to do with whether you can deliver a chemical inhibitor of a much stronger nature to an adult in a safe fashion. And so what we're coming to understand is that our ability to develop therapeutics much more depends on whether the target is approachable, whether hitting this target doesn't have some immediate counter effect in another medical area that's bad, and whether there's really sort of an opportunity with that target. Because with hundreds of GWAS hits, we can afford to be opportunistic about which ones we go after as therapeutic targets. It has much less to do with just how common or just how strong the naturally arising variant is. But what it does have a lot to do with is the fact that we are not nearly as good as we need to be right now at following up the genome-wide association study results. So most of the regional hits in the genome-wide association study point to common non-coding variation that does not, immediately tell you anything about function. In fact, most of the GWAS hits don't even immediately, in any way, shape, or form, link to a specific gene. And of course, very many of the genes in the genome, even if we did have a link to them, we don't know what necessarily the function of those genes is, or the full range of functions that gene has. As I said, most variants are non-coding, so it makes it quite difficult to connect them. And I think that's something technology is being advanced here and in many places that will help make that a more routine and systematic activity. And in many cases, we lack, the validated cellular and animal models that we would need in which to test the function of the gene, and then to ultimately explore potential ways of reversing that particular genetic puzzle piece. So our goal in this activity was not to simply create a nice stamp collection of statistics and colorful pictures for the wall chart, but it was really to sort of inform on the biology of disease in a much more actionable and concrete fashion. And so there's sort of two ways that we have developed approaches and methods over the last couple of years to pursue these questions. And you can sort of just characterize them very loosely as a bottom-up approach and a top-down approach. And the bottom-up approach is sort of a traditional one. If we were studying a Mendelian disease and we finally had the happy day where we convincingly found the gene, then obviously the next step would be we'd study the hell out of that gene, figure out what it's doing, where it's doing it, and which of those potential roles of that or that activity of that gene and variant are on the influential and causal path to developing disease. So we've obviously, especially at the beginning when the number of GWAS findings was small, we're drawn to this traditional way of thinking because, hey, we know that any one of these variants isn't necessary or sufficient to cause disease, but it surely gives us a biological leg up on what's going on in those causal processes. And so one of the first examples of a new, truly novel pathway that emerged from these types of genetic studies was the recognition based on several associations from that first Crohn's disease study of the role of autophagy, which is it's a general process of cellular homeostasis, as being a central one with respect to Crohn's disease. And this was not something that people had been, thinking about or studying, and you can sort of convince yourself by doing a simple PubMed search of that fact. But it has been extremely fruitful over the last decade with Ramnik Xavier and his colleagues out at WashU, Skip Virgin and Pat Stappenbach, developing a tremendous set of experiments that it's really exciting to look retrospectively at through the literature, understanding the functional role of the specific coding variant in ATG 1601, for example, understanding what's going wrong and what's not going wrong with respect to the many different functions of ATG 1601, creating human cellular models and animal models that recapitulate the effect of this variant. They don't recapitulate the entirety of Crohn's disease, obviously, but recapitulating the impact of that variant then allows those to be used as sort of first-line models in chemical screening activities. And so what we've started to recognize is that, a long time ago we sort of recognized that discovering a disease gene was great, but you couldn't just toss it out into the world and hope that a pharmaceutical company knew exactly what to do then. The process that we, now have come to is that in these complex diseases in particular, there's a huge amount of academic science that has to happen first, and we have to sort of meet pharmaceutical development halfway through this process, not just with the name of a gene, but with genes and variants and specific function of those variants and cellular models and animal models that recapitulate the important function of that variant. And so that's a hell of a lot of work, a lot harder than doing a GWAS, I can tell you. So I'm very grateful to have a partner in this activity in IBD, like Ramnik, who's so eagerly working on these things with us for the last decade. The top-down approach is a more conceptual one that, recognizing as the GWAS results started to say that, boy, there's not 5 or 10 genes involved in the processes of disease, there's hundreds, then it rather attracts you to a different idea, which is a little more computational in mind, that shouldn't it be the case that even if there's 100 different associations to genes all over the genome, there should be some coherence to those? Maybe it's not one pathway, but maybe you can find the 5 or 10 critical pathways and processes looking for coherence among those genes. And we and many others have invested a lot of methodologic development into exploring this idea. And some of the most effective ones, I think really are still just beginning to come out, because the most effective way of approaching this, like many problems, would be to have a very deep set of empirical and systematically genome-wide collected data in a variety of other areas, transcriptome, ChIP-seq experiments, transcription factor-based ChIP-seq experiments, and so forth, that you could possibly integrate your GWAS results with. And we're still getting there. But these types of top-down analytic approaches to matching systematic data to other systematic data have really been helpful in understanding that GWAS hits, really do, in fact, in most diseases, point to coherent sets of pathways, and these can be recognized by directly interacting genes in protein-protein networks, to the extent that we measure those and characterize them accurately. They are very helpful in pointing to genes that are, pointing to the key tissues and cells. They're looking at transcriptome experiments that are systematically collected over large numbers of diverse tissues and cells. And with the advent in the last couple of years of ENCODE and roadmap projects and many other projects begin to understand epigenetic marks, we can begin to understand better how these GWAS results, and they do, really overlap with enhancer and promoter elements that are active in specific cells and tissues. So general insights to disease are beginning to come. But of course, general insights to disease, no matter how many times we publish some pathway or processes enriched in disease X, those are not giving us the hard targets we need for therapeutic development. And so we have recognized that our role in gene discovery needs to transition a little bit to a more interpretation of genetic variation that's been discovered, and then also utilizing next-generation sequencing techniques to better target the existing knowledge that we have from GWAS studies to enable us to give more concrete information with respect to the genes and variants of disease so that we can explore those for their therapeutic potential. And so the process, one of the first processes that we've engaged in is really one of trying to fine map the GWAS hits. So rather than just being satisfied with there's something going on in this region of the genome, really zeroing in, looking comprehensively at all the variants in that region, and trying to zero in on what all of the relevant contributing variants and mutations are. And as it turns out, in many of these cases, there's a lot more depth to the genome-wide association result than just a peak on the chart. In the case of the interleukin-23 receptor, this was the first variant that we found in that original study that I mentioned quite a while ago. But as it turns out, we've found 5 total now, so 4 additional genetic variants in this region that all independently contribute to risk of disease. It's not surprising in a Mendelian setting, once you find that this mutation causes disease in this family, you're likely to find other mutations in other families with the same phenotype. Same goes for these types of GWAS results, even though they're much less indirectly causal. And we have developed a suite of methodologies, and Hai Liang from my group has published on behalf of the International IBD Consortium a very comprehensive study recently, fine mapping many of these GWAS hits down to very specific variants and what functional elements they might be affecting. Only half of them, as it turned out, are coding variants, but the non-coding ones are quite interesting, but by and large, we don't necessarily have, other than their proximity to a very plausible gene, we don't necessarily have hard evidence of what the function of those is in too many cases. Putting these types of data together does allow us to get refinements in some cases, and so here's another example from the IBD chart where we have 2 independent hits at the gene called SMAD3. One of them is a traditional low-frequency coding variant. Independent of that, the larger contributor in the region is actually a non-coding variant that we've empirically demonstrated hits a transcription factor binding site that's highly conserved and does empirically eliminate Jun and Phos binding from that particular locus. So ultimately, what we're trying to do for complex diseases is really generate insights and data that allow us to make ultimately a more substantive impact on drug discovery. And so we want human genetics to be routinely informing us not just of, hey, there's a great p-value at RS, whatever, but really to start to give us much, much more specific information about the genes, the perturbations of those genes being genetic variants, in order to create, in the best case scenario, a full genetic picture of what agonism and antagonism of a particular target might do in advance. And if we can build that, and we have to build it not just by looking at each disease in its own little vacuum, but by looking comprehensively across diseases and across human phenotypes, if we can begin to build this more and more routinely, we'll be delivering much more precisely what we promised to deliver when we engaged in this activity. And there are certainly some promising examples. For example, the well-discussed example of PCSK9 inhibition that comes from genetic studies in the last decade, demonstrating that, hey, you've got a truncating mutation that turns off one copy of PCSK9. You, in fact, get a much lower lipid level and a highly significant cardioprotective effect as a consequence of that lower lipid level. As it turns out, there were studies that demonstrated, in fact, individuals with a complete knockout of PCSK9 were, in fact, ultra cardioprotected, apparently, and we'll see if anything bad ever happens to those individuals. But at this point in time, the hypothesis was generated that inhibition of PCSK9 should be, in principle, completely safe and effective. And as it turns out, it in fact, by and large, appears to be both very safe and effective. And so this is sort of the best case scenario, that human genetics can actually pre-validate for you what's likely to happen if you were to develop an inhibitor. And that's really where we're trying to go with the genetic studies now. How do we follow up the GWAS results in order to more generally create these types of full and detailed scenarios that this gene with alleles moving in this direction confers risk or protection to disease. And so that's really where our efforts to use genome sequencing technologies in complex disease have really been applied most specifically in our medical and population genetics community. Obviously, most of the effort, as you'll hear in other sessions, in these studies, again, focuses really on the raw processing and analysis and interpretation of the data, not so much in the operation of the sequencing machines itself, because, again, we've been in a scenario where the technology has just continued to zoom and zoom ahead much faster than our brains and our computers can keep up with it. We are still in the process of learning just how complex the genome is. And Dan MacArthur, who joined us here 5 years ago, has really been spearheading efforts to characterize and create those reference databases that I described that are necessary to this process. And one of the other things that we've sort of come to realize is even though we might now be able to sequence genomes routinely, the power of studies to discover rare variants as contributing to disease are actually even more limited than the power of the studies to define the common variant associations, which is kind of disappointing because it means we'll need even larger sample sizes than those tens of thousands and sequencing is, getting affordable, but it's not that affordable that we can just say, 50,000 cases and 50,000 controls, no problem. Let's churn that out. The sequencing lab would probably be very happy with that, but I don't think we know how to fund that yet. So, of course, much of the focus really resides on using, as I said, prior genetic evidence in a variety of ways, and particularly following up the GWAS hits. And so far, this has been somewhat effective. We've found in our IBD studies, to come back to that, a number of instances where rare variants in a gene that was targeted by the GWAS make independent and much stronger contributions. And so we have several examples that sort of elaborate on this key concept of truncating mutations that are disabling some function or the gene in its entirety being protective from disease. And so it was instrumental that we knew in this early study to focus on this particular gene because it was a GWAS hit. We can now begin to apply this in a more genome-wide setting and generate these types of therapeutic hypotheses that are instrumental to sort of providing a full knowledge base of what might happen if one were to target this particular gene in a very specific way recommended by the genetic studies. So I think I will stop there so we can transition to the next session. The genetic sequencing studies are still at a much earlier stage than the GWAS studies, but they are in fact starting to produce additional examples of this sort of synergy between rare and common variation that we really need to create that sort of virtual genetic-based dose response. And we are excited to continue those. And I am excited that everyone will come back for the next few sessions of this seminar series. So thanks very much. And thanks, obviously, to lots and lots of my colleagues over the years involved in these activities.